Hello everyone, I'm Arpita Banerjee. Welcome to my YouTube channel where I discuss bioinformatics and computational biology. And in this particular video, I'm going to talk about practical examples of post-translational modification from my own research work. This video will discuss proteins that come from pathogens such as worms and viruses. I will talk about those proteins at molecular level and illustrate how they can cause damage to human hosts. You may want to visit my YouTube channel where under the Practical Bioinformatics playlist, you'd find this video called Gene Expression and Protein Modification that attempts to render a comprehensive understanding of the processes within biological cells, including cells of pathogens, that control gene expression in order to control the activity of the cells. And post-translational modifications form an important part of such biological processes that control cellular activity. Post-translational modifications or PTMs may exercise control over the protein structure and function. The chemical modifications could actually render the protein active in certain ways. For those of you who are advanced, you might have come across PTM examples from textbooks or papers or from your own work. And for those of you who are students, you might have just come across PTM examples in your textbooks. Either way, this video could open up a new dimension for you, so stay tuned. Some of the illustrative pictures have been taken from various sources for teaching purposes, and the sources have been acknowledged on the slides. The research examples and associated pictures are from my own publications and preprints. Correlation. Now coming to the central dogma of life, DNA is transcribed into mRNA, and mRNA is translated into polypeptide chain, and you ultimately get a protein. We require the protein for its activity, right? Because proteins run the cell's machinery. Finished polypeptide is chemically modified to control protein activity. The modifications are covalent in nature and can take place either during or after assembly of a protein on ribosome. Amino acid residues may be removed or added, which is rare, or the chemical nature of a side chain may be altered. So essentially, we are talking about two types of modification. One, a chemical group could be linked covalently to the protein via amino acid side chain. This is a list of side chain modifications that could range from phosphorylation to glycosylation to methylation and so on. And the exact residue that undergoes modification is mentioned as well. Here's an illustrative example where you see how the hydroxyl group in the side chain of serine residue could undergo phosphorylation. Threonine and tyrosine residues having hydroxyl group are also capable of undergoing such modification. Residues could be added or removed. Removal of residues is more common, and in number 2 you see how a protein backbone is cleaved to remove residues. Here hydrolysis of peptide bond is taking place. The effect of post-translational modifications could range from conformation change to localization change to chemical property change, and these effects are seen on the protein. This is the sequence alignment of the cysteine protease proteins in Necator americanus, which is hookworm. And now I'll show you some regions and illustrate localization change. This alignment comes from a piece of work deposited in bioRxiv. The title of the work is Cysteine Proteases of Human Hookworm, Necator americanus as Virulence Factors and Implications for Drug Design with Antiheparin and Heparin Analogs, a bioinformatics study. So this is the piece of work, and on the right hand there's a link for downloading the PDF file. This is the first page of the PDF where the author contributions are clearly mentioned. I did this piece of work in collaboration with some scientists at University of California, San Diego. I had worked there from March 2016 to December 2017 for about 2 years, and I finished collecting data for 2 projects in that time period. 2 projects. I am not presenting the entire papers here, okay? Only some excerpts which are relevant to the topic being discussed here, that is protein modification. Now let's get back to the topic which was being discussed. Cysteine proteases. Cysteine proteases are essentially protein degrading enzymes, that is, they're capable of destroying other proteins. These enzymes have cysteines in their active site. This bioRxiv preprint documents the molecular characterization that I did of the 7 cysteine proteases found in human hookworm. Coming back to the sequence alignment to demonstrate how post-translational modification could be associated with localization change. This is a slightly zoomed in version of the alignment. Look at the extreme upper left. You would see a black bar on top demarcating the secretory signal. If you follow the start and the end point of the bar in the alignment, you would get the predicted secretory signal, which directs the cysteine proteases 1, 2, 3, 4, 4b, 5, 6 to the outside. CP7 doesn't have it, and so it doesn't get secreted. Notice the blank space in the alignment. This corresponds to the CP7 sequence of human hookworm. Now, this diagram is again from the scientific article I mentioned earlier. Notice on the extreme left that CP7 remains inside the pathogen, whereas the rest of the cysteine proteases are secreted out into the host extracellular matrix. What do you think these proteins do over there? Remember, I told you that cysteine proteases are enzymes that are capable of destroying other proteins. Well, these enzymes of hookworm degrade host proteins. That is, they degrade human proteins, which is why they are secreted out of the pathogen. What happens after the proteins are secreted out? The signal sequence that directed them outwards get removed. And that happens by cleaving of the peptide bond where the signal ends. So we are talking here about a post-translational modification, that is removal of sequence, involved in localization of the protein. The secreted proteins remain outside after the signal sequence is removed. Though localization precedes the modification in this case, after the modification, the cysteine proteases remain outside the pathogen. Now, what happens after the proteins are localized? Let's take a look at the alignment in figure 1. Again, zooming in, take a look at the proline in the alignment, which is marked by a blue box. Notice the P's boxed in blue. This is the start position of the mature polypeptides. Okay, and on further right, you see the cysteine residues that are boxed within red. That's the active cysteine residue of the cysteine proteases. That is, these cysteine residues are within the enzymatic cleft and they participate in the enzyme reaction. If you look closely, you would see that CP7 has a D instead of a cysteine. That is an aspartic acid instead of a cysteine. So CP7 is not enzymatically active. Also, as I mentioned earlier, CP7 lacks the secretory signal. It's not even secreted out. The motif that you see boxed within purple in CP7 was actually predicted to be a lysosomal targeting pattern. Notice that there are a stretch of amino acid residues between the start point of mature polypeptide and the end point of the secretory signal, right? So even after the proteins get localized to their destination, that is, after the secretory signal gets removed, there's this stretch of peptide that keeps the secreted proteins in immature form known as zymosins. The proteins remain in inactive form in this state. Now take a look at these three-dimensional structures of CP1 to CP6. All of these proteins have the active site cysteine residue and they're enzymatically active only after the propeptide is removed. You see that cleft over here in the protein, a very clear cleft, that's the active site. And the propeptide, when it is present, it blocks this active site. Therefore, the stretch of amino acid residues between the start point of mature polypeptide and the endpoint of the secretory signal, shown by the red bar here, gets enzymatically cleaved off, which results in the conformation change of the zymosins, and then they turn into active cysteine proteases capable of degrading other proteins. So here's another example where cleavage of protein backbone is involved in converting immature zymosins into active protease structures. Now I'm going to talk about an example of covalent modification, where the side chain of an amino acid of the protein gets modified, and that could result in chemical property change. In this particular example, I'll talk about covalent binding of heparin to the cysteine proteases of hookworm. Heparins are glycosaminoglycans that include molecules such as hyaluronan, chondroitin sulfate, heparin sulfate. In this particular image, what you see is the chondroitin sulfate attached to the core protein via serine. The linkage to serine is covalent in nature. In case of hookworm cysteine proteases, the covalent linkage of heparin is via asparagine residue. In figure 1 of the mentioned manuscript, you will find a region demarcated as N-glycosylation motif region. This is where the heparin molecules are predicted to bind. This is zoomed in version of the alignment, and if you see at the N-glycosylation motif region, at the start position, you can find the asparagine or the N residue in most of the cysteine proteases. This is where heparin molecules gets covalently linked to the enzymes. When the cysteine proteases leave the pathogen, when they're secreted from the pathogen, they already have heparin molecules, at least most of them have heparin molecules bound to them. The heparin molecules are indigenous to the worm. What purpose do they serve? Why are those heparin required in the first place? Heparin in bound form acts as a cofactor to the enzymes and possibly exercises allosteric control over the enzymatic action, making the NSCPs functional even at alkaline pH, which is required for the survival of hookworm that encounters different pH conditions in its life cycle within the host. I further postulated that upon release of heparin molecules from the cysteine proteases by deglycosylation enzymes present in the small intestine of human host, heparin possibly serves as a cofactor to antithrombin, which inhibits thrombin by binding to it, thereby interfering with the coagulation cascade, leading to blood thinning of the host. When hookworm sucks blood from a human host, it would not want the host blood to get coagulated. Coagulation would mean interference in blood sucking, and therefore the pathogen actually targets the coagulation cascade of its human host. Protein phosphorylation is one of the most important modifications that lead to a diverse array of biological response, such as metabolism, hormone action, cell growth, and therefore cancer, gene expression, memory, etc. As mentioned earlier, the residues that primarily get modified are serine, threonine, and tyrosine because of the hydroxyl group they have. Enzymes that carry out phosphorylation of proteins are known as protein kinases, and the enzymes that carry out the reaction in the reverse direction, that is, enzymes that dephosphorylate proteins, are known as phosphatases. Now, for the purpose of showing you a real example, this is the profile alignment of N protein or nucleocapsid protein of Chandipura virus isolates with other representative rhabdoviruses. Alignment comes from one of my publications, Whole Genomes of Chandipura virus isolates and comparative analysis with other rhabdoviruses. It's an open access paper. This is the first page of the PDF of the article, and you can see that I and Rashmi S. Gunjikar contributed equally to the work. Rashmi was actually part of the sequencing team, whereas I had done the bioinformatics analysis of the sequences. Speaking of rhabdoviruses, you will see rabies virus in the alignment. This Chandipura virus work had been done by me at National Institute of Virology, Pune. I had worked there from October 2007 to April 2010. For about 2.5 years, I got 2 publications from the work I did at the institute, and I'll be presenting relevant parts from one of my publications. Going into the paper, you will find the profile alignment, which I'm going to talk about a little. This profile alignment of N protein has sequences of viruses belonging to family Rhabdoviridae, genus Vesiculovirus, and genus Lisavirus. Profile alignment mode of cluster X was used to create this alignment, where profile 1 was made up of sequences consisting of genus Vesiculovirus, that includes Chandipura virus, and profile 2 was made up of sequences of genus Lisavirus, that includes rabies virus. If you notice in the phylogenetic tree, you'd find that the vesiculoviruses are distantly related with the Lisaviruses. The purpose of the profile alignment was to glean information from other well-characterized viruses that would not otherwise align very well with the comparatively newer and somewhat distantly related Chandipura virus. Here, in this part of the alignment shown within the blue oval are the putative CAM phosphorylation sites in the N proteins of the viruses. The motif residues are in bold font, and if you notice the motif in case of CIN 6514, it begins with a K or lysine, whereas the other representative Chandipura viral isolates CIN0327 and CIN0728 have a R or arginine. Though K and R are somewhat similar in the context that both are positively charged, but nonetheless, they are different amino acids with different properties. Now, why does it matter? See, CIN6514 isolate comes from the first reported Chandipura viral outbreak in Maharashtra that happened in 1965. Back then, the virus was not causing encephalitis in children, whereas the other isolates are from outbreaks from other geographical parts in the years 2003 and 2007, and these were causing encephalitis. So the virus had definitely evolved to be much deadlier. And it goes without saying that most changes happening at the gene level translates to the protein level and has the potential to contribute to final biological outcome. In this context, it is imperative to mention that the mutation is appearing within the N-terminal region containing the first 47 amino acid residues, which have been shown to be critical for self-assembly of nucleic acid protein. Now, there's another such example in this part of the alignment. If you look within the pink oval, you would see the predicted CK2 phosphorylation sites in some viruses here. Upon closer inspection, you'd find that a motif for CIN6514 or the isolate from the 1965 outbreak is SRED, and the motif for other Chandipura isolates are SRDD. So we have a E or glutamic acid which is getting replaced by D or aspartic acid as the virus has evolved. Again, though E and D are somewhat similar in the context that both could become negatively charged, but nonetheless, they are different amino acids with different properties. In this context, it is imperative to mention that the deadlier Chandipura viruses are accumulating the mutation at a place which in case of Lisa viruses has been shown to be critical for viral RNA transcription and replication. Also, deriving from the alignment, the region could be delineated as an important T-cell epitope. More importantly, the mutation appears in a region which has been shown to be important for n-RNA interaction in Chandipura viruses. Now, in both the cases I mentioned, the exact residue which undergoes modification is not changing. Speaking of the CAM phosphorylation site here, if you look at the Chandipura viral isolates, then threonine is the residue which could undergo phosphorylation. But that remains the same. What changes is some other residue in the motif. Now, turning our attention to the CK2 phosphorylation site, if you look at the Chandipura viral isolates, S or serine is the residue which could undergo phosphorylation. That remains the same. But what changes is some other residue in the motif. So how does it matter? See, the binding of kinases or phosphorylating enzymes depend on the flanking residues or the motif residues as well. So even seemingly insignificant or small changes, such as a change from K to R or from E to D, has the potential to impact the level of phosphorylation of N proteins and hence their functioning, making the proteins from the deadliest strains different from the prototype. ScanProcyte is my go-to tool for motif prediction, as ProCyte is a database of protein domains, families, and functional sites. You can search ProCyte by motif IDs or by sheer name of the motif. You can even scan a protein of your interest for predicting putative motif. I'm really thankful to Sarah ma'am who gave me an opportunity to work in her lab at National Institute of Virology. And I'm also thankful to Vidya ma'am who provided me funding for work. I hope you find the content useful and I will make the next video when I can. Thank you very much.